So thank you so much for the introduction. And first of all, I would like to really thank the local organizers of this event. This is my third year here as invited lecturer. And every year I really enjoy the workshop, and also Olomouc is a very nice city. So it is really a pleasure for me to be here. So, first of all, for who does not know me, I would just like to present myself. I'm Federica. I'm associate professor at the University of Naples, and I'm come from the. Okay, so sorry. I came from the biomolecular simulation drug discovery laboratory with my team, and two of them are here in the audience. And basically, what we are doing in our lab. In our lab, we work basically on ligand target recognition, and we study both ligand target against protein with unbiased molecular dynamic simulations, maybe sometimes to study sometimes some conformational changes within the receptor or also within the nucleic acids. But sometimes such kind of interaction need to be studied with enhanced sampling methods. So we are doing also biased molecular dynamic simulation. This is one of an example of funnel metadynamics in which we can enhance the ligand target recognition within its target by enhancing the sampling, because you have to know that ligand target recognition is called as a rare event. So we need the time scale of microseconds or seconds to be happen. So sometimes ligand target recognition can occur. We can see this process only with enhanced sampling method. But today we will start with the basic overview of this kind of approach, because I will show you just this is a topic of the lectures, and just we will start with some basic principle about molecular docking, and then we skip to the current docking methodologies and what are the strength and also the weakness of docking, and how we can enhance the molecular recognition or the scoring with artificial intelligence. Then at the end of the talk, I will leave you some take-home message, because I think that the most important thing of this workshop is to go home with the basic thing that you have learned in this workshop. So let's start with some basic principle of docking. Okay, every cellular processes happens between two molecules interact between them. This is really driven, this is a process that is driven because some interactions occur between a molecule and a target. And basically, we can have two kinds of interaction, attractive forces and repulsive forces. And the equal balance between attractive forces and repulsive forces make possible the interactions, because we know that the ligand within a target can go everywhere in the surface of the protein. So, but not all the surface of the protein are can accommodate the ligand. Okay, so in this sense, repulsive force help us, because when this kind of forces occurs, the ligand cannot bind in the target. But when attractive force win against the repulsive force, the ligand will prefer a cavity, a binding pocket within the target, and so the interaction can occur. So this is what happens at atomistic level, and this is a process that we call molecular recognition. So given a target and a ligand or a drug, we can it can be established some molecular interaction between them to form a complex. Yes.
You just have to click.
Okay.
Now it should be working.
Okay. So, okay, so we have just talked about the interactions. Okay, so the importance, of course, in the attractive interactions like hydrophobic, van der Waals, hydrogen bond, pi-pi stacking interaction to make the complex, to lead the formation of the complex. But they are not only the leading factor during molecular docking recognition. So also the geometric and the shape complementarity is really important in a docking procedure, because when the surface of the ligand can match, can fit also the cavity of the binding site, then we can have the best thing to accommodate the ligand. And so this is a very nice question that we have to think about. On Monday, we have learned the Professor Kirkmer lessons, the importance about the shape of the ligand in ligand-based virtual screening. I will add that the shape of the ligand is important also in docking, in structure-based, because when they fit, the shape of the binding site and the shape of the ligand fit, we can, of course, have a good binding. So the first question we can answer is, how can we reach the right ligand conformational sampling before the docking can occur? And we know, in fact, that when the ligand is in the unbound state, the ligand can undergo to very different conformational change. So the first question we have to answer is the reaching the right conformational sampling of the ligand to enter in the binding cavity. Can we answer to this question? We will see later if we can answer to this question. So let's skip now some docking methodologies. There are, of course, many types of docking methodologies. The first one is the blind or global docking. This kind of type of docking occurs when we don't know exactly where the ligand can bind the target. So we do not know from experimental point of view where is the real binding site. So the best thing we can do in this case is to build the box large enough to accommodate the entire protein surface and include all the possible binding site within the protein. Of course, this is always a good thing to do, but there are some problems, because we have to run several docking procedures to identify the correct binding sites. And this can lead to some errors in the results. So when you perform blind docking, be careful, because you are not interested in finding the right pose of the ligand, rather than we have interested in finding the right place, the right binding sites. So
Takže se můžeme zase potom přesunout zpátky.
There is some question from the audience.
Ne, já vím, že, ale jestli potom coffee breaku
What is?
na tou semena, ať už je nahoře.
A nebo že bychom tam
nebo
There is some microphone.
To je ale další ztráta. Ale my to navíc musíme doladit.
Hele, tak až to bude zamít do fungující na světloce, tak v té chvíli přijď jako a my uvidíme, jestli to bude ještě v rámci Fedorky.
I don't know.
I'll try to make them.
Okay.
Okay, nice.
Is it okay? Let's go. Okay, so this may introduce some error in our calculation. So to do docking, blind docking, by thinking about which is the right binding site and not which is the right binding pose. Then we have the targeted or local docking, and in this case, instead we know from experimental point of view which is the right place, so the right binding site. And in this case, we have to build the grid box. It is enough to build the grid box large enough to accommodate the real binding sites. So in this, I suggest in this case to put the centroid of the grid box in the center of the binding site. So it is quite a good thing. Now, of course, in this case, the docking accuracy is based in the input information of the binding site. We will see later which is the importance of have a good quality model for doing an accurate docking procedure. So other types of docking. Other types of docking are rigid docking and flexible docking. What does it mean? In rigid docking, we have the classical theory proposed by Emil Fischer, which is the key and lock. So we think the target and molecule as both rigid. So only the ligand which fit the right shape complementarity of the binding site can go within the binding site. So this again show the importance of the shape complementarity. Then we have instead the flexible docking, called also as induce fit, in which we introduce some flexibility of the binding site residues. So the binding site can undergo to some conformational changes in order to adapt themselves with the shape of the substrate or the shape of the ligand. So there are, I think, a little more accuracy with respect to the rigid docking. Okay, so now I would like to talk about something that sometimes is really omitted during docking procedures, and it is the role of water. Water is everywhere in biology. Okay, so binding sites, protein binding sites are full of water. And this is something we think about because sometimes the presence of water within the binding sites can influence docking procedure. In fact, when we have water inside binding sites, water actually is considered as a competitor of the ligand binding. And what is the reason of that? The reason is that basically we have unhappy waters and happy waters. What does it mean? Sometimes we can have molecular waters that can prefer to stay in the solution rather than to stuck into the binding sites. So these are energetically unfavorable, and we need to displace them during the docking procedure. And by displacing them, we can have ligand with a big potency, for instance, during a virtual screening. But at the same time, we have also happy waters. So we have waters that are so-called as functional. So they contribute to the ligand target recognition. And in this case, they contribute to the specificity of the ligand recognition. So in this case, we have to leave them. So, and when we have this kind of approach, we have obviously to think that the presence of water can lead in a change of the shape of the binding site. For this reason, it is strictly correlated to the specificity. So the question is,
Again.
yes.
Should be working.
Okay.
Is it working?
Okay. So the question is to leave or not to leave. Should we leave the water or should we delete them? I will answer by saying that we have to be careful about the thermodynamic properties of such water molecules in the binding site. I would like to show you just a really nice example that I taken from Professor Leffer about this kind of virtual screening. So in the left panel, we have a co-crystallized ligand, the original co-crystallized ligand with these two unstable water molecules within the binding site. And then we have in the right panel, we have a screened ligand, a screened hit that perfectly match within the binding shape, and this ligand has higher potency with respect to the previous one. So in this is a very nice example in which by deleting some waters, we can have a ligand with a higher potency. So we have to think about that. And so the final answer is calculate the thermodynamic properties of those molecular waters and remove those waters that are unhappy. And I think that when we have a situation like that, so you have downloaded the crystal structures and you analyze the binding site and you see some waters inside, I think that the best way, the first thing to do is the water map, that is a very nice software of Schrödinger, and to calculate the thermodynamic properties of the waters. So let's go on. We have talked about the quality of crystal structures, so go on to speak about that. And so another thing that we have to consider during a docking procedure is the model, the protein or nucleic acid model. Is it of good quality? Do we have co-crystallized structures? So the best thing is obviously to have experimental structures like NMR or X-ray structures. But sometimes we do not have experimental structures. So in this case, we need to the homology model technique. And of course, it is not the best thing, but we have nowadays we have some validation procedure or enhanced models that can help us with homology model. So let's say how we can see that the crystal structure is good or it is bad. First of all, we have to look at the electron density map. So pay attention to the resolution of the target And for instance, here we have some residues with some of them with a very bad resolution. And here in the lower panel, we have two examples of good quality crystal structure. So as we can see here, the electron density map in these two kind of example well define the shape of the residues, especially in this D panel, rather than the first two panel in which we have an undefined shape. So we do not know which is that residues. We cannot have information about the amino acids. So I think that the PDP educational portal is the right thing that help us in this case to choose the model with the highest resolution. Another thing to do is the preparation of the target. To prepare the target means that we have to sometimes correct the connectivity and to add hydrogen atoms because in PDB model, especially in crystal structures model, we don't have all hydrogens inside. So another thing to think about is how can we add hydrogen atoms? How can we treat protons? The best way, the answer is we must predict them as good as possible. And this means that we can not only add all the hydrogens, but we have to consider also the tautomeric form, okay, in our case. So let's skip to the homology model. So when we do not have a right crystal structure, homology model is a well-used technique that it is based on the fact that sometimes we can predict the three-dimensional structures of a model by looking at the similarity rates of their sequence. So sequence that are similar can have we can use their sequence to predict the model and to add, for instance, and to predict the quaternary or tertiary structures of a protein because there is the principle that models with the same sequence are in high probability to have the same fold. And actually, we have a lot of homology model websites that help us to do a very right homology model. Of course, we have to validate it. I suggest you to validate your model when you do homology with, for instance, molecular dynamic simulations. And this is true, of course, for proteins, but also for nucleic acids. So when we speak about nucleic acids, the things is even more complicated. So there are some limitations because sometimes the sequence of two proteins can diverge significantly, and so we have they cannot be detected by standard homology model. In this case, we have the help of a enhanced prediction model. So for instance, AlphaFold, as we can see nowadays, is very widely used to efficiently predict the right model, so with the very high similarity with respect to experimental one. Now let's skip to another question for docking procedure. So we have spoke about the ligand conformational sampling. So how we reach the right conformational sampling of the ligand? This is the first challenge of the docking, because when we think to a ligand, we need to think that the ligand undergoes to translation and rotational movement, and especially when it is in the unbound state. So, and depending on the orientation or the changing of the torsional, the ligand can undergoes to some basins, energetically stable basins, or some hills. So you have to think about the ligand conformational sampling as a potential energy surface, which is made by basin and hills. The hills are energetically unfavorable conformations, while the basins are the lowest one, the lowest energetic conformation. So the good thing is to reach a conformational of the ligand able to be stacked into the basin of the potential energy surface. So we need to think in the problem of reaching about lowest conformational states. And this is also a big problem in docking, because when we put the ligand in the binding site, of course, we will have a sort of entropic loss, because some movements and some torsional or rotational movement of the ligand cannot be occur. So we have to think about that. So this is the main challenge, the first challenge of docking, of course. And so this is an example of a ligand with several rotatable bonds. And for each rotatable bonds, we will have some basin and some hills. And so depending on the degree of the torsion of the rotational movement. So we need to take only the most stable conformational sampling. And this is very important. The sampling of the ligand is very important, because the docking program will use that conformational that will explore all the possible conformational space, and on the basis of the conformational space, they will rank the final pose. So the possibility of having the right conformational space is given by the Boltzmann equation. So the probability of a ligand to be found in a certain state, for instance, into a basin, is given by the Boltzmann factor and the partition function. So it is inversely, the probability is inversely proportional to the potential energy of the ligand. And what does it mean? It means that low energy states are more heavily populated than the high energetic states. So my suggest is to identify conformation within 1 or 5 kcal per mole from the global minimum, and then these states will represent the highest probabilistic states in the Boltzmann population. And we have a lot of course, conformational sampling, systematic search, random search, and of course, molecular dynamics. These two are the most widely used. Random search, in the random search, especially with Monte Carlo or genetic algorithm, we will perform random conformational changes of the ligand, while of course in molecular dynamics we have the most accurate conformational sampling, but also it's computationally expensive. So once we have spoke about the search algorithm in the docking, we now skip to the scoring function, because depending on the conformational sampling and the pose that each docking software will reach within the binding site, of course, each program will rank them. So we will have a good score or bad score of the ligand. And this depends in the type of the scoring function that is included in the docking software. Actually, what are scoring functions? Scoring functions are mathematical equations, and that describe the way in which the ligand pose, is recognized as a good pose or a bad pose. And we have basically three kind of scoring functions that work in different way. For instance, we have the knowledge-based scoring function are basically depend on the will take the statistical information from the PDB complexes, and basically it will use a sort of a grid box. It will collect all the possible conformational experimental PDB structure. They collect it and build of each of it a sort of grid box. And the grid box is, we can imagine that the grid box is divided into grid points, and every grid point is related to a probe atom. So which is the frequency that an oxygen atom is found in that position, or nitrogen atom is found in that position, or carbon atoms is found in that position. So we have a collect of a frequency of the grid points, and with the Boltzmann statistics, we can convert the frequency into the interaction energy. So when we use docking software that work with the knowledge-based, of course, we are interested in reproduce the right experimental structure rather than we are interested in the calculation of the binding free energy. Force field or physics-based instead, for instance, Autodoc or Gold, works instead by summing the non-covalent energy terms that came from the natural force field. For instance, Autodoc takes the information of the non-covalent terms from the Amber force field. And so they work basically by adding the van der Waals parameters or the Coulombic terms in the calculation of the final binding affinity. And also they include the H bonds over the van der Waals and the Coulombic terms. They include also the H bond and the desolvation terms. So they there is a sort of they are more close to the physical significance in the binding affinity. Then we have the empirical instead. The empirical scoring function approximate the ligand binding free energy because it's simple sum up the contribution of a number of individual terms like lipophilic, Coulombic terms, also the rotatable state that with the ligand internal energy are seen as the entropic loss. So we it is added also the entropic loss in the torsional and the rotational movement of the ligand. Of course, they are much faster. They are much faster than with respect to the other scoring functions. But the risk is that to have a discrepancy between the binding affinity and the real activity of the top ranked score. So to sum up, what are the mechanics of docking? Of course, very important are the search algorithm to search the right conformational sampling of the ligand, and of course, the scoring function. When we have to choose which search algorithm or which scoring function, so which docking program we have to use, we have always to make a compromise between speed and accuracy, because sometimes speed does not mean accuracy. Okay, so we have to choose. And the right balance between the scoring function and then the search algorithm will affect, of course, the docking assessment. So the accuracy of your ranked pose and which pose to choose to be sent to biological experiments. So let's see strength and weakness of classical scoring function or classical docking. Okay, now we have talked about the mechanics of algorithm, of docking, of course, and now we have to choose. Now that we know how docking works, now we have to choose which is the best recipe. So which kind of scoring function, which kind of search algorithm I should choose. And why one may think, okay, the game is simple. I will perform a random search. I will, for instance, choose the force field based. So I will choose autodock, for instance, or gold, for instance, and then I will get the best molecule ever. I will say the top 10 ranked pose, ranked ligand, and I will have the best drug ever. So I can send to clinical trials. This is not right. You should think that docking never tells the whole story. Okay, so binding affinity does not mean binding activity. The top listed molecules you will have, independently for which scoring function or which search algorithm you have performed, you will never have your top score, your top poses as the best ligand. Okay, so ligand with good score doesn't always mean a good binder. The risk of have false positive is high if you make this kind of analysis. Okay, so why this? For two reasons. The first reason is that it is not possible to sample the entire conformational space of the ligand in a small amount of time. Okay, so this is the problem of the speed. Okay, we cannot be sure that independent, even if we choose molecular dynamic simulation for our sampling, the conformation we will have, they are not, we cannot have a convergence of results. The second reason is that there is a bias of the scoring function. What does it mean? That different software tools use different scoring method. We have just seen before. So the same ligand, we will have very different score depending on which software you have used. So for this reason, we can have, this happens, I think, always, false positive. So it is not enough to choose, okay, I will choose the best 50 ligands and I will send to biological test. This is not a right thing. So how can we solve this problem? The best thing is, when speaking about classical scoring function, is to take as much as information for each scoring function. So I will add as other scoring function the consensus score. Which is the consensus? We will speak about consensus and the importance of having consensus. So docking procedure cannot guarantee the success in vitro screening. So the ideal model is that, okay, we have performed this virtual screening with some thousand of molecules, then we calculate the AUC, area under the curve, and so we have very good area under the curve, so I will send my best molecule to biological test. And all of them, they are active. This is an ideal case. But in reality, it's not the case because molecular recognition process is a very complicated process, and scoring function make approximation in the calculation in the binding affinity. So a possible solution to this problem is to compare all the scoring function and to weight good scoring function with bad scoring function. Okay, this is called the consensus score, the consensus scoring function. I think there is this very nice paper published in 2018 in which they show the importance of the consensus. So, which is a consensus? In consensus docking, we have several targets, for instance, but we can have also one target, for instance, and several molecules. Let's say that we have to perform a virtual screening. And for my virtual screening, I should use not one docking program, but several docking programs. Okay, so I will perform the same screening with within the same binding site with the program 1, let's see, Autodoc, for instance, program 2, Glide, for instance, and then program, whatever you want. And then I can rank the score of each program, and I will merge all of my results. Of course, I will have different results. It's mathematically, I will have different results for each scoring and for each program. And then what I can do is to calculate the exponential consensus ranking, the ECR, in which the probability to find the right molecule is given by the score reached by each ligand. So it is a way to weight a ligand that have a good score with the program 1 and the same ligand that have a bad score with the, for instance, program 2. So, okay. So let's skip to how we can improve the scoring with artificial intelligence tools. First of all, I would like to a little bit speak about this slide because I found a very interesting slide. This is a work published 2 years ago, and I think this is a clearly overview of how artificial intelligence identified molecules can have a high rate of success to enter in clinical trials. If we see from 2015, the growth of molecules entered in clinical trials exponentially growth, and in 2023 we have a total
So, can you hear us now? Yes, now it's possible to hear us.
Okay.
So, this is your microphone.
Okay.
Sorry for that. All the time.
No problem, no problem. So, just want to look at this second panel, which represent the discovery mode. We can see that in 2015, most of the molecules were repurposed molecules, but now most of them are small molecules, so new entities. And I think it is also a very nice is of significant is significantly because of course this means that artificial intelligence tools can have hit writing success because they are a robust model, not only in predict in ranked the pose, the docking pose, but also to predict the ADME properties, the toxicological properties of a molecule. In fact, if we look at the okay, so it's not it doesn't work. It does not work. Okay. I don't know. I think there is something wrong.
I think it was the code. Yes.
Okay. Okay. When we think about the phase 2 trials, the success rate of molecules is of 40%. That means that probably, I think that beyond the business thing, I think that maybe AE should improve. Now there is also the prediction of biological activity. So this is still a gap. Okay, so dogging is not enough. We have already seen why, because of the search algorithm failed to achieve a good full convergence of Leon sampling, and also between because the scoring functions, both physics-based or empirical-based, are suffering from reliability issue. So we need the help of deep learning or generative models, AE, tools, because they can also dramatically reduce the time and the cost of early phase drug screening, but they can also have a good prediction both in the scoring position and also in the generated best pose. So we can just have two kind of models. We have the deep learning scoring and the generative pose prediction. The first one is based on the sampling, is a sampling based method. For instance, DiffDoc, SafDoc, Nina. There are just few examples, but of course we have many other software, and they simply rank the pose that maybe we have obtained with physics-based method. And then we have, of course, a generative pose prediction based on the regression-based model, in which they work by generate a new pose within the binding sites. For instance, Karmadoc or Carsidoc are some example about that. I would like to just spend 2 words about Nina, which is an algorithm that is also included in the software that we use in the tutorial session. So just a few words about Nina. Nina is a fork of Smina, which is a fork of Autodoc Vina. So it is an improvement of the scoring of Autodoc Vina, and it is A-based because it improves the rank of the scoring of your molecules. It is based on graph, so both ligands and the target are treated as graph. We have already seen in the lecture of Professor Kickmar what are the graph. And once they have treated this graph, the problem is, the fact is that Nina built around the complex grid box, and the grid box is divided into some kind of pixel that are known as voxel, and each voxel represents a chemical features. For instance, there is here the feature of hydrophobic, the feature of hydrophobic, of hydrophilic points, or electrostatic, or something like that. So it works by learning the patterns of interaction by doing a message passing neural network. And basically, it alternates pooling and convolution convolution neural network. In the pooling, there is keep only the essential information about the patterns of interaction, and then this information is treated by the convolutional neural network to learn the essential interaction. And this is alternate between pooling and convolution, and this alternance will ensure the quality of the predicted, the ranked poses. At the end, Nina will produce two kinds of score: the CNN score, which is referred to the pose quality, and the CNN affinity, which is referred to the binding affinity. However, whatever tools you will use as enhanced scoring, I think that there is a very nice work published last year, and in which we can see here that with respect to some physics-based, for instance, here glide, all the A-scored based sampling and also generative model are able to predict good quality of model with an RMSD lower than 2. And it is important also, this second slide, in this second panel, we have the quality of the prediction referred to some kind of random results. So the brown barrier means are the PV value. That means, okay, this result is random or it's a robust model. As we can see in, for instance, in KarmaDoc, we have equal to 0 PV value. So this means that KarmaDoc as a generative model can produce a good quality, a robust model, a good quality of the pose, and it is robust. It is not random. Okay. So I think that whatever software you will use, of course, you can enhance your scoring prediction. So, final take-home message. I think this is the most important slide. Please, when you perform docking procedure, think the docking procedure as a starting point, not the final answer. Okay, then think about the scoring function. Scoring functions are powerful, especially physics-based, are powerful, but they are still from reality. So there is not so a good accuracy between the binding affinity and the real activity of the molecule. There is a big gap. So don't trust just the score number. the number is just a number. So you have to visually inspect your poses, even those that are worst ranked. Use a refinement scoring or generative models to increase the prediction, improve your prediction. But think about that AI scoring functions does not replace physics-based, but complement it. So use this tool just to improve your classical docking results. And of course, once you have performed all of these analysis, the best thing is to validate your model, of course, with molecular dynamic simulation long enough to see conformational change, maybe. Always keeping in mind the experimental data, if you have experimental data, or keep in mind what you want to obtain from your molecule, which kind of activity you want to have from your molecule. So thank you for your attention, and I open for the questions. Other questions? Please.
Thank you. I would like to ask about docking to complexes with DNA, because you discussed mainly docking to proteins. So please, could you comment on docking to DNA, if possible, and does it work?
Yeah. Okay. So, yes. Performing docking with nucleic acids, of course, the scoring function or search algorithm are the same, obviously, but it is even more complicated than the protein, because in DNA you have, a well-defined binding site, but they are most solvent exposed. Even if you consider, for instance, double helix DNA, the major groove or the minor groove are mostly solvent exposed. So there is an high risk of having discrepancy in your results, because there could be loss of accuracy in the desolvation prediction calculation. Okay, and this is also true for instance, the non-canonical structure, for instance, the G quadruplex. So in this case, for the case of G quadruplex, is even more complicated, because basically you have to do a blind docking most, and so you have to run several docking to see, and molecular dynamic simulation to see if the binding you have predicted is right. So I think you have to think about the role of the solvent maybe more with respect to the protein.
Thank you.
No problem. Other questions?
Okay, that is good. You save a little bit time. You can ask questions during the coffee break, during the lunch, so Federica is still here.
Yes.
What I would like to say, I received a notification that the previous lecture room is now working, so we can move there.
Okay.
I hope that everything will be fine. So let's move. We probably will cancel a coffee break. We will see. Sorry for this. Was probably on this computer and make a recording, so we should still have the video.
I would like to know more about the guy, but
No,
the basic and the good old things that finally when AI doesn't have data, I guess anyway you can.
I have a question about the enhanced sampling. To the start, you showed the binding and unbinding. So I just want to ask, what methodology are you using? Is it just umbrella sampling, or you enforce force there? And what is meta exactly?
also, I sampling, but the Gaussian potential within your system that should remain underway from the frequencies. And so you can add this potential.
So here you simulate the unbinding as there is a potential towards the filter.
The potential started with the complexes. Then I
And which software are you using to add the Gaussian potential? Are you using what software, like Gromax, OpenMM? What? Oh, I don't know that one. Med? Okay, I will. Okay. Thank you. Never heard of it. Sure.
Please, could I follow up with my previous question?
Sorry?
Please, could I briefly follow up from my previous question?
Oh.
Thank you. In my case, I'm trying to model a complex of topolymers 2, which contains DNA and protein. And my task is to also rationalize activity of some compounds, which should be intercalators. And based on that, we think that they plot this complex of DNA and the enzyme. And what I did, I used machine learning like AlphaFold 3, fold the system together. But I would like to add like one more method which could like shed a little bit more light if the AlphaFold prediction has some sense of it, or if it's like kind of hallucination. Okay. So do you think that
actually is? I think it predicts quality of also DNA. I have used once. And it gave me a very interesting results because it predicts, the